The third method for predicting the genetic outcome of the genetic process is using probability. What is probability? It is a study to find out the chances or possibility of an event to happen or occur. In this case, let us take a fish A, whether it will be able to feed on its prey or it will not be able to eat. So what is the event is, it is able to eat or not able to eat. Okay. So finding out the possibility, chances of whether A will happen or B will happen. So predicting which event will happen, call this probability. Okay. So it is the likelihood of an event to happen. Event A will happen or B will happen. At what frequency? Whether A has more chances or B has more chances, or only A will happen and B will not take place, or both A and will happen. So what is the chances, the possibility of these two events to happen? How is that we are using probability for predicting the outcome of the genetic process? Here if you take, we are going to use two rules. One is the multiplication rule of probability and the other rule is the addition rule or sum rule of probability for predicting the outcome of the genetic process. If you take this product law, it is where we are dealing with two independent events. So when two independent events take place, and here when we want to calculate the probability that both the events will take place at the same time, how do we calculate is by multiplying the probability of these two independent events. And the second is the sum law. Here, probability of a generalized outcome. If this outcome is by more than one way, then the probability that A or B will take place, how do we calculate, is the sum of all the individual probabilities of all such events. See, overall, if you take the product rule tells us what is the possibility of both A and B to occur at the same time. Whereas the sum rule tells us with the possibility of A will occur or B will occur. We will see this in detail. We will see that in detail in about this multiplication law and how we are using it for, why we are using it for the genetic process. If you take in the multiplication law, we told if the two events are independent events, then the probability is found out using the product rule. So what is this independent events? So two events are happening and the outcome of one does not affect the outcome of the other event. So such events are called as independent events. For example, okay, let us take event A is tossing a coin and event B is rolling a dice. So the outcome what we can get from tossing a coin is, it can be a head or a tail. So that will be the outcome. But whereas in the event B, the outcome will be getting a number 6 or number 5 or number 2. So if you are tossing a coin, the outcome in the dice is not going to be affected. And rolling a dice does not affect the outcome of whether you get a head or a tail. So these two events are independent events. One outcome of A does not affect the outcome of B. Okay, that is happening of A does not affect the happening of B. How do we relate it to our genetic cross?
In the Mendelian inheritance, what are the independent events and why we are taking it now, considering it as independent events, and how we are using the product law. Let us take a cross, genetic cross, where you are crossing two heterozygous parent, and when this from a parent, when the gametes are formed, okay, the alleles get segregated. That is what we learn in the law of segregation. So what are the independent events? What is the event here is, event A is of gamete getting a dominant allele is event A, let us take. And the probability, the other event B is gamete getting the recessive allele. So two events here. Let us take these are two events. Gamete getting a dominant allele and gamete carrying a recessive allele. So two events. And when we learnt about the Mendelian inheritance, we know that this segregation of these alleles are independent events. That is, a gamete getting a dominant allele does not affect the gamete getting the recessive allele. So these are the independent events. Okay, so they are independent event. A gamete getting a dominant allele probability is 1 by 2, and the gamete getting that is a recessive allele as a probability of 1 by 2. And where do we use this product law?
Since these two events are independent events, the chances of both the events occurring at the same time, for predicting that, we are using the product law.
Formation is a combination of these gametes. Okay, so you can combination of this gametes, which is equal to the probability of both the events occurring together. Both the events, what is this? A gamete carrying capital D and B, both occurring together. Combination is this occurring together. So we are finding out the probability. What is the chance of both the events occurring together, which will give us the zygote formation. So here, since the gamete formation is an independent event, the probability of the zygote formation for predicting, finding out the probability of the zygote formation, we are using this product rule. Where product rule, what we are telling is probability of getting both event A and B at the same time. So if you want to find the probability of both A and B at the same time, we use the product. How do we get is getting a that is, you are multiplying the probability of event A with event B. That is what we are doing in genetic cross here to find out the zygotes. So here, combination of the alleles, okay, D and D. So you are multiplying 1 by 2 into 1 by 2. Okay, I am writing it here. It is 1 by 2 into that is equal to 1 by 4. So the chances of getting both dominant allele in the zygote is 1 by 4.
So this is what is that is probability law, and how we are using it in our genetic cause for finding out the outcomes. The second law, what we are using here is the law sum law or additive law. What is the sum law, and how is that we are using it for genetic cross, we will see. What is the sum law or sum rule? The sum rule is applied for two mutually exclusive events. What are these mutually exclusive events?
are two events which are disconnected. That is, only at a particular time, only A can occur or B event can occur. In the previous case, independent event, we were finding out what is the probability that both A and B will occur. But here in this case, mutually exclusive is either event A only can occur or only B can occur, not both at the same time. So such events are called as disconnected events. Mutually exclusive events are two events that cannot occur at the same time. For example, let us take event A is today is Tuesday, and event B is today is Friday. So here today can be either Tuesday or it can be Friday. Not both A and B. Today cannot be Tuesday and Friday together. Events are disconnected events. Both cannot occur at the same time. Such events are called mutually exclusive events. So where are we finding mutually exclusive events and using this probability sum rule of probability? When we are seeing some rule is applied here, when we are trying to find out the probability of just having the dominant triad in the F2 generation. So here we have seen that we worked out the gametes and what is their probability, and we saw the combined phenotypes, combination of both these alleles occurring at the same time. What is the probability? So later after this, what we want to find is what is the ratio of this, what is the phenotypic ratio and genotypic ratio. So if you see here the phenotypes, what you get from the F2 generation is, what are the phenotypes? Here you get a tall, and the other phenotype you get is dwarf. And if you see the frequency, at what ratio this tall and dwarf is occurring in the F2 generation. So if you want to study that, if you see tall, okay, what is the frequency of tall? We see that we get dominant triad from three events. And all these three events are considered to be mutually exclusive. That is, if one event takes place, the other event cannot take place. Since all these three events are mutually exclusive, we are using the additive rule or the sum rule here. So to get the probability of this tall plant, okay, that is in multiple ways we are getting this tall plant. So we are adding up all these events, probability of these events. That is equal to 3 by 4. Similarly, when we are talking about the genotype ratio, we will be having the homozygous, where you will be having only 1 by 4. Okay. And the recessive also you have 1 by 4. But here, if you take the heterozygous dominant, you can get it can be from two events, either from this event or from the other event, where you get the dominant allele from the female parent and the recessive allele from the male parent. The other event is where the dominant is from the male parent and the recessive is from the female parent. So you get the heterozygous genotype from two events. So when you want to find out the probability, you go for using the sum rule. Therefore, 1 by 4 plus 1 by 4 is equal to 2 by 4. So therefore, if you see in the overall event, only 1 by 2. So the chance of getting the heterozygous is 1 by 2. The probability of getting a heterozygous dominant is 1 by 2. That is what is explained here. So what we were talking, why we are using product rule, why we are using additive rule, all that we saw. So this slide is explaining you from the beginning. That is a heterozygous parent. When the gametes are formed, so since we know that it is following, it is that is segregation is an independent event. Okay, so these are two independent events. A gamete getting a dominant allele does not affect the other gamete getting the recessive allele. So they are independent events. Since they are independent events, to find out their combined, so to find out to get their combined probability, here we are using the product rule. The zygote, the combination of these alleles in the zygote, we are using the product rule. And again here some rule where we are using is when we want to find out the outcome, that is whether it is a tall progen. When we are talking about one outcome, that is this tall triad. Here this is from many events. So here each event is a mutually exclusive event. Therefore we are using the additive rule. In dihybrid cross, we know that segregation of alleles from two different locus are also assorting independently. So we will use the probability law here. A gamete getting a segregation of alleles of a particular triad, of triad A, is also independent. Similarly, the alleles of triad B also segregates independently. So that is what is represented here. A gamete getting capital R and gamete getting small r is 1 by 2 and 1 by 2. And similarly, if you take here, gamete getting capital Y is 1 by 2 and small y is 1 by 2. So using a branched diagram, we see the gamete proportions here. Gamete getting, so here if we want to find out the combination of combined occurrence of both these alleles. So we are using the product law, that is 1 by 2 into 1 by 2. That will be 1 by 2, where you can get gamete with R and capital Y. So the other gamete which will carry capital R of capital R allele and small y allele of the other gene. So the probability is 1 by 2 into 1 by 2, that is equal to 1 by 2. Okay, so similarly the gamete proportions are given here. And when we go for the F2 offspring, so this gametes from the female parent is given on the x-axis and the male parent is given in the y-axis. And here we're finding out the combined occurrence of the gamete carrying capital R and Y and the other gamete carrying capital R and Y. So the zygote which is having both these occurrence. Okay, so a combined occurrence of these two alleles. So is by how do you find out here is by multi-product rule. So 1 by 4 into 1 by 4, that is equal to 1 by 50. So similarly, for all the boxes we have worked it out.
So tri-hybrid cross, this we have worked in the class. Tri-hybrid cross, also you can use the probability, and here you use a branch diagram which will be easy for finding out that. Okay, so take this as an example and work it at your home.